Good afternoon. My name is Kimball, and today I'm going to talk to you about how machine learning, automation, and computational biology go together at scale. First, I'd like to give you a bit of brief background about myself. Throughout my career, my goal has been to find interesting problems where big data can lend an advantage and build solutions. My career started much lower down in the stack of the infrastructure layer at Cloudera. There I worked on Apache Hadoop, made some Hadoop training materials, and also developed the Apache Scoop project. Scoop helps you import data from relational databases into HDFS. In 2010, I founded a company called WeBeData that helps organizations build big data applications with a particular focus on the e-commerce sector. Oops. Oh, God. Yeah. Okay. And for the last year, I've been the CTO of Zymergen. Zymergen is a software and data company revolutionizing bioengineering with a combination of machine learning, automation, and data-driven process improvement and search techniques. And we take this capability and focus it on the problem of optimizing the genomes of microbes that produce chemicals. Later in this talk, I'll get into more detail about exactly what this means. But at a high level, I would like to observe that Zymergen is a business built on positive feedback loops. These feedback loops are both internal and external and help our business move forward. But moreover, I don't believe that these loops are particular necessarily to our business or even our industry. I believe that they affect and can apply to everyone in this room. The first of these loops is an external loop, and I'd like to observe that innovation happens faster in open industries. On the right side of this slide, you can see a cycle wherein the falling cost of enabling technologies enables more people to use these technologies, which further drives down the cost. But there are other industries, in particular software is one of them, where open community discourse helps propel innovation forward faster. And by community discourse, I particularly mean the ability for technical individuals in different organizations or institutions to communicate clearly and unambiguously about deep technical topics. This might require a particular open standard or a particular open technology, such as open source software. And businesses that can align their technological paths along technologies that serve with community discourse lead to higher degrees of innovation. The second loop that I'd like to talk about is an internal loop to Zymergen. And this is our perpetual quality improvement loop. At Zymergen, we are fanatic about quality, and we are really fanatic about data. So we're always collecting more and better data on our processes and our results. When we analyze this information, this allows us to make better predictions about what's going to happen in our factory workflows. It allows us to dig deeper into failures that occur along the way, to analyze them and smooth them out of our processes. And it allows us to perform data-driven workload optimization and determination. At each cycle through our factory, we take a pause and determine what are the most efficient things to push through this factory in order to serve our next set of goals. This overall leads to greater confidence that our methods work, which is important when you're pipetting around colorless liquids, and it optimizes the throughput and runtimes of our system. By leading to higher throughput, we are always collecting more and better data, a larger corpus of data on our processes and their results. And I'd further like to observe that virtually every step in this cycle is enabled in some way by big data systems, and in particular open source. Tools like Hadoop and Spark allow us to process information at scale. Open source machine learning toolkits allow us to make predictions about the future. And various storage technologies allow us to integrate all of the different sources of complex data that we need to get our jobs done. So to get deeper into what Zymergen actually does, we make industrially fermented chemicals. What does that mean? Well, everybody knows how fermentation works. Leads to some very tasty beverages. But in addition to producing alcohol, microbes, when fed feedstocks like sugar, can also produce all sorts of other compounds. These can be commodity or specialty chemicals, paint components, cleaning detergent agents. They can also be agricultural products such as enzymes, fertilizer additives, animal feed enriching agents, amino acids, artificial flavors, fragrances, therapeutic drugs, and more. Industrial fermentation is one example of a broader industry, bioengineering. Industrial fermentation takes on a few different basic forms. One of these is the production of ethanol for biofuels. It's an additive to gasoline. In other cases, we can take advantage of chemicals that microbes produce naturally. Microbes produce all sorts of different chemicals, but they typically produce them in trace amounts. In the before picture on the rightmost side of this slide, you can see that maybe 1% of the input energy associated with a microbe is being devoted to the orange chemical. If you change the DNA of this microbe, you can optimize it so that 40, 50, or 60% of the energy in this microbe is being devoted to producing a chemical that we want for industrial purposes. But we're not just necessarily limited to the universe of chemicals that microbes produce naturally. We can also change the metabolic pathway of this microbe entirely by replacing or adding large components of its genome, which allow us to produce a broad variety of chemicals that would never be found in nature. Bioengineering also has applications to the medical sector. One of these is industrial fermentation-based. Many drugs, such as penicillin, are created through industrial fermentation. But we can also do really interesting other things with genomic editing. For example, gene therapy is one potential cure or aid for various chronic illnesses. Some patients who have cancers and certain types of cancers have this because of defective or missing genes in their genome. By taking a blood sample from this patient, changing the genome to reintroduce a healthy copy of this gene, you can then load this DNA sequence into an inactivated virus and use that as a delivery vehicle to add these genes back into the genome of the patient to help them get better. In biological computation, there are other examples of how industrial fermentation can help. Bioengineering can help. Biological computation borrows a lot of different analogies from electrical engineering. Circuits, switches, latches, oscillators, these terms all have analogs in the space of DNA. So we can use DNA editing to create sophisticated biosensors that can detect trace amounts of heavy metals or other toxins in an environment and can serve as an early warning system for people who work in hazardous conditions. The point of all of this is that DNA construction is becoming a routine engineering effort. This graph, which has a logarithmic y-axis, shows a few different things. The blue line is Moore's law, the density of transistors on a chip. The red line shows our efficiency at reading or sequencing DNA. And the yellow line shows our efficiency going up over time at being able to construct new DNA sequences. This is becoming cheaper by orders of magnitude, literally, over the last few years, and faster. But while creating any particular genomic sequence is becoming easier and easier to do, there's still a wide open problem. What is the best DNA sequence to serve some particular goal, like, say, maximizing the amount of a particular chemical that a microbe might produce? To get DNA optimization right, you need to do two things. First, we need to vastly improve their modeling capability. Genomes are a very complex, high-dimensional space, and as humans, our intuition about where we might go looking in this genome to affect a change simply isn't well calibrated. And second, we need to, in a wet lab environment, test changes physically a lot more efficiently. And in particular, by efficiency, I mean doing this with high throughput and with high precision so that we can detect small amounts of change or improvement in the trait that we're trying to optimize for. At Zymergen, optimization is at the heart of what we do in order to create microbes that make chemicals. Our workflow has 4 broad steps. In the first step, the design phase, microbiologists will use computers to design a set of genomes. Each of these genomes takes the current genomic sequence of the microbe we're working on and includes one change in each of these different genomes where we want to make a test. In the second phase, we build the microbes that actually have these genomic changes. So we might come out with 1,000 different microbes in a tray, each of which differs in one small position in its genome from its neighbors. We then test these microbes, feeding them sugar and seeing how much of a chemical they produce. And in our final analysis phase, we incorporate all of this information into our next round of genomic designs, concentrating and aggregating the results of winning changes in the genome, and also looking at failures and other side conditions that might be arising, and using those to direct our future course of events as well. In order to optimize genomes, we need to test a lot of genomes. And to do this efficiently requires that we test things in parallel. On this slide, you can see a pipette, which is the basic workhorse tool of a microbiologist working in a wet lab environment. Can you imagine producing 1,000 genomes with this pipette? That would take a lot of time. Fortunately, though, we have better tools. This multi-channel pipetter can work on 8 wells at a time. Of course, why stop at 8 when you could have a 12-well multi-pipetter? Or actually, if you wanted to really go crazy, they make 48-well multi-channel pipetters as well. So now you can get a decent amount of throughput by hand. But fundamentally, while certain industries have made a great stride in incrementally improving a small hand tool, this really isn't going to take Zymergen where it needs to go. For that, we need to look to an entirely different place, and that place is robotic automation. Automation raises the bar for consistency, quality, throughput, and efficiency that we can perform our genomic changes and testing with. On this slide, you can see 2 different robots that we use in our factory. On the top, you can see a robot arm that moves plates of DNA from one workstation to another. And on the bottom, you can see a liquid handling robot. This liquid handling robot has a 96-well multi-channel pipetter in the middle, which certainly improves our ability to do things in parallel. But it also improves the consistency and auditability of our work, making sure that we track every touch and change that we do in our laboratory. But once we've got automation down, this brings about some pretty big analytic challenges. First, if you could construct 1,000 genomes at a time, you'd need to propose genomes to construct 1,000 at a time. Then we need to track these workflows through our laboratory. And we need to correlate our results with the input genomes using these in a feedback loop. Helix is Zymergen's in-house software for DNA design and analysis. And by baking in Helix and our automation systems, we've created a software-defined factory. The various bubbles in this workflow are performed either by humans or by automated laboratory robots. The edges in this graph are performed by Helix and driven by our software. We manage the work orders from the design phase to the building phase, track the workflow through the building and testing steps, collect all of the information at the end of that test cycle, and then push this through a series of automated analyses. There's a few basic functions that Helix performs. For example, there's a lot of quality control. Biology is inherently probabilistic, which means that every time you perform a step on a set of DNA components or genomes, some of these are going to fail. So we need to continually track what is still successful and what would constitute a valid test result to carry forward. We need to calculate microbial performance over prior generations. Does this have an increased throughput in chemical production? This is really hard when microscopic changes in our factory can cause major sources of error to accrue. And finally, we could forecast failure rates and future construction probabilities. The more complicated a change that you want to make to the genome, the higher the probability that it's going to fail, so the more factory resources you have to devote to developing this one particular change. So we have to make a continual set of trade-offs between the complexity of changes that we make and the number of discrete changes that we can make. All of this is done in service of optimizing areas of interest in the genome. So whenever we've found locations in the genomic code that have caused some change up or down, we then spend time trying to understand what would be the best change to perform in this area. As I said earlier, the space of DNA is very high dimensional, and so we have to use statistical sampling techniques to ensure that we've made a good coverage of every area. Helix has to perform a lot of data integration steps from a variety of different data sources. On the right side of the slide, you can see an image of cell colonies growing on an agar tray. Each of these cell colonies was created from one individual genomic change, but there are several such colonies available on the plate. We use image analysis software to determine which of these colonies are sufficiently far apart from their neighbors that we can actually pick one up and deposit it back into a well full of liquid. The ones that we can pick up are color-coded in yellow on this slide. We also store this information so that we can track the growth rates of different cells with different changes and understand how that might affect the phenotypes or traits that we're trying to optimize. On the right side of the slide, you can see a screenshot from a software as a service tool called Benchling. Benchling allows our scientists to dive into the genomes, understand them, and also mark them up. The lines that you can see inside that circular plasmid of DNA each represent an annotation, a region of the genome that's been highlighted by a scientist and been given some various parameters. Some of these are notes to their colleagues, but others are directives that we read in our software to use as inputs for future genomic changes. As I said earlier, quality control is one of the key areas that we have to continually optimize in our facility. On the right side of this slide, you can see Sanger sequencing output. Sanger sequencing is one mechanism by which we can determine what's the precise set of bases, A, G, T, or C, that occurs at each position in the genome. The peaks in this graph represent the confidence that at each particular location we know what the actual base is at that location. So if we're trying to match up whether these set of peaks match to the genome that we expect, where do you put the thresholds to balance between the false positive and false negative rate? Since all of this costs capacity in our facility, it's something that we continually pay attention to and tune. And finally, Helix provides a powerful set of genome design tools to our scientists. The old-fashioned mechanism of specifying precisely and literally which genomic changes that you want to make doesn't scale if you have to devote resources to designing thousands of genomes at a time. So we provide declarative tools that allow scientists to, say, take cross products of different libraries of promoters or other genomic components and attach them in series to each of the different locations where they might be applicable in the genome. We then construct all of these in turn to determine which are the effective locations. There's a number of different open source tools that are also attacking various areas of computational biology at scale. Atom is a genomic computation library built on top of Apache Spark. It provides data structures for the description of DNA and other complex related data types, as well as algorithms for parallel read alignment and other algorithms that you frequently want to perform at scale. This is being used at Berkeley for a project for the petabyte scale storage and analysis of human genomes, and it's all available as open source. As I mentioned earlier in this talk, having alignment with your industry in an open feedback mechanism allows innovation to be driven forward faster. Labs as a service present a really interesting adaptation on this front. Labs as a service allow you to consume laboratory resources incrementally on a per lab protocol basis. Much like EC2, you rent and buy what you use. Instructions are delivered to these labs in a machine-agnostic format. These instructions could be carried out by a human who pipettes things from one place to another, or a variety of different liquid handling robots. You mail them the physical specimens that you want to operate on and email them a protocol. They execute that protocol and send you back your data and will mail you back physical samples from your experiments. This allows researchers around the world who might have no access to a wet lab whatsoever to design genomic changes, design a protocol to test those changes, and get results back. All of this is pushing forward the degree of innovation in the bioengineering industry. So to wrap things up, as I've termed it earlier, I believe that the software-defined factory is the new software-defined networking. Taking flexible components in a factory facility that can be reconfigured on the fly and individually optimized to increase throughput, lowers costs, and accelerates the business forward. And this efficiency and flexibility is driven by a combination of automation, big data, and machine learning. Furthermore, it dovetails very neatly with our devotion to continuous data collection and continuous process improvement. And finally, I'd like to just mention again that open community standards and open source have helped us and help others lower cost, let anyone process large-scale data, run experiments, and enable innovation. I'd like to close with two shameless pitches. First, we're hiring. And second, we're hosting a happy hour this evening, and we'd like to meet all of you there. It's going to be at 17 Maddox Street from 5 to 8 p.m. My name is Aaron Kimball. Once again, thank you for listening.